Hi, everyone. Welcome to Spatial Omics Seminar Series 7 again. And today we have our second speaker of the seminar series. And Professor Jerry Chen will be talking about neural network physiology and spatial transcriptomics interface. But before he gets started, briefly let me introduce him. And Professor Chen received his PhD in 2010 from MIT. Then after his PhD, he did a postdoc at the Brain Research Institute at the University of Zurich. And then around 2016, he started his lab at the Boston University in the Photonics Center, Department of Biomedical Engineering, and Neurobiology and Department of Biology departments. And currently his lab is focusing on the question around how the brain assembles sensory information into abstract representations. And for that, they are investigating long-range neocortical networks. And with this, I'd like to turn over to Dr. Jerry Chen.
Great. Thanks, Ahmet and Ron, for the invitation. It's great to be able to speak to you all today and talk about the work that we do. Seems like we have a very diverse audience, in terms of people who are working either in neuroscience, but maybe outside of neuroscience. And so I thought I would kind of, give a little bit of a kind of a broad perspective of some of the questions that we're interested in studying and how the use of spatial omics or spatial transcriptomics has come particularly in handy. And it's be great to kind of, talk to you guys more about, potential ideas or where things are going and the questions that we could potentially try to tackle with these types of new technologies that are coming online. So let's slide here. Okay, so so just to start with a kind of overview, so our lab is a neuroscience lab, and we're interested in studying cortical circuits, underlying behavior.
So excuse me, everyone, can you put on mute? You are not talking.
Thanks. Yeah. So it'd be great if.
So Jerry, can I mute?
Can you hear me? Okay.
Yes.
Okay. Is there do you hear anything in the background, or is it coming from somebody else?
Yeah, I hear. Maybe I mute you and.
Yeah, the background seems to be fine right now.
Okay. I turned on the air conditioning somehow this morning, so I don't know if that's what you're hearing or if you're hearing somebody talking.
Sorry, I think that's my side. I think it's okay now.
Okay.
I apologize.
Okay, no problem. All right. Let's see. All right. So yeah. So the questions that we're interested in studying is how circuits, carry out functions that are related to things like perception, decision making, goal-directed behavior. And we think about the brain as circuits. And in order to understand the circuits, one of the important types of information that we need to understand are the individual circuit components. And so that relates to the idea of cell types or neuronal cell types. And many of you probably know that not all the neurons in your brain are all the same. They're quite diverse, and they can potentially carry out different functions, right? And cell types in general are defined by anatomical features, such as their morphology, how they're connected together, also in terms of their functional properties. A lot of these types of properties are really defined or informed by the molecules, right, that are expressed in individual neurons, which can be quite diverse and specialized. And so when people think about cell types, they think about cell types a lot in the terms of transcriptional states. What are genes are being expressed that defines the function of that cell and within the circuit. Now, when we think about transcriptional states in the brains, it's important to keep in mind that certain transcriptional states are dynamic, right? So under the context of neuronal plasticity, learning and memory, there are sets of genes that are dynamically regulated with response to activity or experience that kind of go up and down, and set potentially wiring patterns or changes in properties that allow for new things to be remembered and learned. Also, it's good to keep in mind, right, that transcriptional states are dynamic over the course of lifetime of an animal. During development, right, certain gene programs turn on and off that define the wiring of the system, and they kind of, maybe more stabilize as an adult, but it continues to progress, as through aging and so forth. And so when people think about cell types, at least in the context of adult animals, they're thinking about markers that are largely thought to be static or stable across time. And recently, there's been a large effort to define or catalog all the types of cell types there are in an adult mammalian brain using single-cell RNA sequencing methods. And through these methods, one recent studies, such as those carried out by the Allen Institute, have identified on the order of maybe 300 or 400 different types of putative cells, cell types within the cortical nervous system. And so this is, now that we're starting to gain a better appreciation of the diversity of cell types that are present within the nervous system, the question now is, how are these particular types of cells interacting? What are their particular functions within the context of behavior and information processing? A couple types of principles, just for people to be aware of, that have been identified are a couple of things have been identified. So, right, there are excitatory neurons. About 80% of the cells in the cortex are excitatory, 20% are inhibitory. If you start to look at the types of cell types that constitute inhibitory cells, what people find is that based off their gene expression profiles, you see that inhibitory cell types can be largely subdivided in a hierarchical manner into largely non-overlapping discrete cell types. And these types of cells are generally common across different cortical areas within the brain. They might be abundant in different proportions, but by and large, they're found throughout the brain or throughout the cortex. In contrast, when we think about excitatory glutamatergic neurons, we see different principles. So excitatory cell types can either be discrete types of cells, or they can form gradients defined by combinatorial patterns of gene expression. And the genes that are defining a given cell type in one cortical area can in fact be different depending on the different type of cortical area that you're looking at, whether you're looking at a motor cortex or in a visual cortex. So there are some differences between how the genes are being expressed between excitatory and inhibitory neurons. Now, the current methods for neuroscientists to identify and study the function of these cell types in vivo in awake behaving animals are somewhat limited. So historically, one of the main methods for studying different cell types is through the use of transgenic animals that will label using one particular type of gene, defining a cell type, expressing a fluorescent molecule that one can then either isolate or target to perform recordings on, for example. Another method that's also been used is through imaging technologies I'll talk on in a second, where you can image cells in the awake animal and then post hoc find those same neurons and perform immunohistochemistry looking for protein markers that are defining these particular types of cells. Both of these have limitations, right? One of the main limitations is that these labeling strategies only label one gene at a time. And so when we're looking at these genes, these cell types, you can really only potentially look at one or two cell types within the brain. You can't necessarily look at how multitude of cell types are potentially interacting and communicating to each other within the context of a larger network. The other limitation is that a lot of these markers are defining, not only specific cell types, but families of cells, right? So we are not necessarily looking at, when you think about a parvalbumin neuron, this is not necessarily a given cell type, but constitutes, one branch, of the large hierarchical tree, which are composed of many different types of parvalbumin and positive neurons that each have different sets of gene expression patterns. And so to really be able to really look at a specific cell type, you really have to not only look at just one marker, but combinations of different markers. And so this was where, things like spatial transcriptomics has really taken off, and really recent years has really opened up the possibilities for being able to not only look at cells with higher level of genetic precision identity, but also being able to look at how different types of cells are interacting with each other, communicating with each other within the population. So this brings us to a different idea, or an idea that I think is evolving, which is now that we have the ability to kind of look at these things in a multi-omics way, it really is changing the way that people like myself and others who do systems neuroscience can approach certain problems. And so I think, historically, a lot of the ways that we try to understand circuits by looking at behavior, neuroactivity, connectivity, or gene expression has been carried out in what I consider like a horizontal approach, where you study behavior in a given set of subjects, maybe neuroactivity in another set of subjects, gene expression in another set of subjects, and then kind of through combining these different informations, you can try to put together a picture of how circuits work. But what I think is an important avenue moving forward, and which some of these technologies are enabling, is a more vertically driven approach, in which within a single individual animal, we can start now to look at both behavior, neuroactivity, connectivity, gene expression, and all within the same animal to really look at how individual animals and their circuits are really carrying out specific computations in the brain. And so this is kind of a modality that people in my lab are trying to corral around and really push forward and and making use and leveraging to understand how the brain works. So we focus as a model, understanding how information is being processed using the mouse Whisker system as our model system. And so many of you might not be familiar with the mouse Whisker system, but mice use their whiskers to really feel and touch their way through the environment. So they kind of walk around the environment, they move their whiskers, and they have the same or a comparable level of tactile acuity using their whiskers as we do, for example, with our fingers. And there's a really nice organization in the cortex where we can start to study how information is processed in a given whisker and monitor how that information flows through the entire nervous system and gets expressed or computed within the cortex. So there's this nice, what people refer to as a barrel-like organization of somatosensory cortex, where you have individual whiskers, corresponds to a population of neurons here that will encode information specific to that whisker. And so in addition to this organization of primary somatosensory cortex, this area is also highly connected with areas like motor cortex, which controls the whisking behavior of the animal, other areas like secondary somatosensory cortex, which starts to encode more higher level features, things like potentially object-like representations. And so this is a great system for us to understand how information is being processed, how the animal is really engaging with the environment to understand the world it's sensing. So we have developed behaviors that will allow for different types of sensory processing, tactile sensory processing, and I'll kind of walk you through this type of behavior that we're trained animals to. So we use microscopes that I'll talk about in a second that will allow us to image neuroactivity with cellular resolution. And so for us to do these types of behavioral experiments, we have trained mice to be head fixed or stationary underneath a microscope, so we can get really high resolution images. And in this particular type of task, this is kind of like a tactile working memory task in which you have a rotor here that will come in and will deflect the whiskers in two different directions, right? It can deflect a whisker in an anterior direction or a posterior direction. And the trial is designed such that the animal is being subjected to one set of stimulus, so the rotor will rotate in one direction or the other. Then I will pull back for a couple seconds. and it'll come back in, and then we'll either rotate in the same direction or a different direction. And the animal is trained to lick to get a water reward if the two stimulus are different, and withhold licking if those two stimulus are the same. So I'll kind of play out a video of how the behavior works. Right, so here's the trial starts. The rotor comes in, it will rotate deflect the whisker in a forward direction, come out for a couple seconds, come back in, rotate in an opposite direction, and the animal realizes these two sets of stimuli are different. It will lick to get a water reward. And then the next trial I'll show here is it's what's called a matching trial, in which the rotor rotates in one direction, pulls out, comes back in, and then rotates in the same direction again. The animal knows there's no water reward, it withhold leaking, right? So this is a very powerful type of behavior because it really allows us to look at different aspects of cognitive function. You can look at how information is being processed, sensory information. The animal is required to essentially use working memory. It has to retain the initial bit of sensory stimulus it gets for a couple of seconds. And then as the second stimulus comes in, it has to retrieve the prior information, make a comparison. Then it has to be rule, that the animal has to mentalize whether or not things were the same or different, right? Equal or not equal. And then the animal has to then report what that set of stimuli were in order to get a reward. So it's a very rich behavior that we can really study a lot of aspects of cortical function. Now, the parts of the brain, as I mentioned, that we're interested in studying are the somatosensory cortex, but also things like motor cortex that controls the whisking or the licking of the animal. And so we're using a lot of technologies that will allow us to monitor activity across large populations of neurons. And for those of you who are not familiar with how people record activity patterns in the brain, maybe many of you are familiar with like using electrodes, recording electrical activity. But one of the forms of recording activity that's very highly compatible and lends itself to things like spatial transcriptomics is calcium imaging. So when neurons fire activity, calcium enters the cell. And so we can use genetically encoded calcium indicators that will fluoresce in response to this calcium influx. And this is operates as a surrogate for spiking activity within populations of neurons. Right? Since it's a fluorescence type of sensor, we use 2-photon calcium imaging microscopes that allow us to image activity in the brain non-invasively. So we usually have to remove a part of the skull to provide optical access, but we can then put a glass cover slip on top, essentially giving us a window to the brain. And the animals can be performing behaviors, and you can use our microscopes to literally look at the brain, look at activity in individual neurons, the populations of neurons, and understand how the population of activities is being encoded during behavior, right? So this is one video from a collaborator, Spencer Smith, who has developed one of these microscopes that we call a mesoscopic 2-photon system. So it's different from standard systems in that it offers us a large field of view. So our lab is heavily involved in optical engineering, and we have taken his design and kind of modified it and built our own microscope systems that will allow us to image activity across different cortical areas to understand how these parts of the brain communicate with each other. So we have this very large microscope that allows us to image activity in sensory cortex and also motor cortex simultaneously, and that will allow us to look at the interactions and communications between brain areas during behavior. Now, because this is an imaging-based approach, and because we can, shows you an example of the field of view. This actually shows you an example of the type of implant that we use. It's called a crystal skull, in which we put a window on top. And so what you're looking, and it's sealed with bone cement, so the animal is perfectly fine running around. But again, we can peer in this we have using viral approaches express our genetically encoded indicators. And if you zoom in using our microscope, you can see individual cells, and you can record all the activity patterns, which you see on the right here, from these different areas. All right. So because we're actually looking at the cells and we have spatial information from our neural recording, this really lends itself to spatial transcriptomics. So what we wanted to do was to leverage this capacity for functional imaging and then be able to ask, what are the types of cells that we are imaging, right? Based off the genetic expression patterns that others have identified using the RNA sequencing work that they've done. So the technology that our platform that we developed is called CRAC, which stands for Comprehensive Readout of Activity and Cell Type Markers. And I'll show you this animation for how it works. So we're imaging activity in the brain of a behaving animal using our 2-photon imaging system. And once at the end of our behavioral experiments are finished, we can take the brain out and then we can isolate the tissue and we can find the same neurons that we imaged in the wake animal in our fixed tissues. From then, we can take that same tissue and then we can perform HCR FISH to stain for given sets of genetic markers that I'll go into a second that will help us to identify the types of genes that are being expressed that define the cell types that we're looking at. And the really, I think, amazing thing for me that's developed in recent times is the ability to multiplex, which we can stain for a given set of markers in one round, strip using DNA, and then restain for a different set of markers. And so you can across multiple rounds, we can build up gene expression patterns that we need in order to decode what type of cell we're looking at, and then be able to correlate that cell type information again with the neural activity of the neuron. Okay, so just because there are a few probably experts in spatial transcriptomics in this audience, I thought I would just briefly kind of discuss some of the logistical or methodological reasons for why we picked this particular set of approaches in this kind of first iteration of the CRAC platform. So one of the trickiest things, or one of the most involved things when you're going from an awake imaging an awake animal to going then into tissue is being able to find the same neurons, right, between an awake animal and then in the tissue. So this aspect of ex vivo to in vivo registration is a critical part for the platform. And invariably, the way that we are imaging, we can image in vivo across different depths of tissue. And to increase the likelihood of being able to find the same neurons in tissue, it's become apparent to us that the more that we can work with thick, intact tissue ex vivo, the easier our job it will be to find the same neurons, right? So I know a lot of technologies currently are working with things on the order of 10 microns, 20 microns. That makes it quite difficult sometimes to find exactly the same neurons, or they would be existing across multiple tissue slices. And so we really wanted to focus on tissue that's relatively thick, about 150 microns thick sections. So because a lot of technologies so far as I've seen, kind of are more optimized for thin, very thin tissue sections, we try to pick ways, kind of robust ways for us to be able to stain and image the 150 micron thick tissue sections. And so early on, the things that we looked at that seemed to work best was a combination of tissue clearing, so hydrogel embedding of our tissue, which allows both better optical access to be able to image across the 150 micron thick tissue using confocal microscopes, as also well as the hydrogel allows for easier penetration of the HCR FISH probes. We went with the HCR FISH probes because it was commercially available, seemed kind of off the shelf ready, easy for us to use, who are not experts in these particular technologies. And the only constraints or concerns that we had when we try to use thicker tissues is to be able to reliably register not only the cells, but potentially individual single mRNA molecules. And so we do have mRNA resolution since we're using spinning disk confocals, but we were a little concerned given these kind of thick tissues, whether or not would we really be able to reliably identify the same mRNA molecules. And so we took a little bit more of a conservative approach that I'll talk about in a second, in that we went through a design in which we really just wanted to pick out markers that we could really relatively easily identify as expressed in a yes or no manner within individual cells, and where we didn't necessarily have to rely on being able to multiplex in a manner that kind of goes into individual single mRNA. Although that would be great, and it'd be great to talk to you guys more about ways where we can kind of do this type of work in more thick tissue. So this is an example of the type of staining scheme that we're using. So our first application of this approach was to focus on the cell types that exist in layer 2-3 of the Whisker barrel cortex. And so we picked out on the order of about 16 markers and leveraging what was already known from the single cell RNA sequencing work, we picked out markers that we were confident or had high confidence that would be largely non-overlapping markers and strong markers that span the types of cells that we wanted to study. So we picked markers to identify 3 of the main excitatory cell types in layer 2-3 that I'll talk about in a second, as well as some subfamilies of inhibitory neurons and their subbranches here, which I'll also talk about in a second. And so through the selection of these 16 markers, then we program a barcode readout where we can, stain for multiple markers within a given round, but doing it in a way where we have some error detection to be able to kind of resolve or make sure that a given cell was in fact staining, a given marker or stain for a given marker. And we did this across 2 imaging channels. And I'll show you what this data looks like. So this is an example of what it kind of looks like after registration across 6 rounds. We're usually using our genetically encoded calcium indicator which is protein the mRNA, to actually kind of re-register the same cells across multiple rounds. But hopefully you can appreciate that markers that we're using are relatively strong markers that sometimes are maybe so abundant that you're not necessarily seeing single molecules, but there are some other markers that as far as we can see are properly capturing single mRNA molecules. But the big thing is that they're more or less binarily expressed within one cell type and not in a different cell type. So it's just more in a matter of just looking and making a yes or no call. Does the cell express something or not in this particular given round? And then we can decode exactly what type of gene it was expressed and the type of cell that we're looking at. All right, so I've kind of talked a little bit about the methodology for how we're doing the spatial transcriptomics, and I'll talk now about the data analysis and some of the things that we found. Okay, so one of the things that we need to do is characterize the activity patterns as it relates to behavior in these animals. And so we're using generalized linear models that will try to capture or predict the activity of the animal of the given neuron with respect to aspects of behavior. We focus first on some of the types of inhibitory neurons. And so one of the types of neurons that we focused on was the somatostatin neurons. And so somatostatin neurons can be broken down into subfamilies. One of the families are those that express the marker CHODL, and those are distinct from other SST neurons that don't express CHODL. What's already known about the neurons that express CHODL, SST, are that most inhibitory neurons are local neurons. They don't necessarily extend their axons outside of their local cortical area. However, these SST CHODL positive neurons are actually unique in that they have these long-range axons, and they're also interesting in being, they seem to be pretty active in sleep. And so we wanted to know, well, how do these types of neurons respond when the animal is performing a task compared to other types of SST neurons that don't? And one of the things that we find or confirm is that when we look at the ability of SST neurons to encode aspects of like the direction of the stimulus that we were providing in animals, what we find is that the SST neurons that don't express CHODL are highly responsive to tactile stimulus, but the ones that are CHODL that are positive are not. And again, these are the ones that are associated with sleep. So we can already see these type of distinctions between how different subfamilies of these inhibitory neurons are responding during task behavior. Similarly, if we look at the VIP subfamily of inhibitory neurons, these can be broken down into those that express PTHLH and those that don't. These are also correlated with other markers that people have been studying, such as VIP neurons that express CCK or calbindin. And what's known about the ones that express calbindin, the PHTLH positive neurons, are that they have these very long bipolar morphologies, which are different from the VIP neurons that are PTHLH negative, that have a more of a basket multipolar morphology. So already there's some anatomical differences. On a behavioral level, what we find is that the PTHLH negative VIP neurons are more responsive to Whisker stimulus, and whereas the ones that are PTHLH positive, these bipolar neurons, are more responsive when the animal is whisking in air. So and that suggests potentially greater motor inputs or different types of responses that combine more motor-related information. So we can already also see these types of breakdown in functional properties as you start to break down these trees of branches of these inhibitory neurons. Okay, so those are some initial findings that we have from the inhibitory neurons. For the rest of my talk, I'll talk about what we see in the excitatory neurons. So as I mentioned, we focused on 3 types of excitatory neurons in S1, ones that are called ADAM-TS2, BAS1A, and the third is AGMAT. And just to think about, what these excitatory neurons are doing in sensory cortex. So it's thought that the excitatory neurons in sensory cortex have the ability to capture both so-called bottom-up and top-down information. So bottom-up information is essentially the information related to the world around us that we're bringing into our brain. Top-down is the inferences or the context that can shape the bottom-up information. So these are things that are learned, that provide our own subjective experiences to the sensory information around us. So for example, if I'm looking at a traffic light, I see the color red, right? Red is like the external information that's coming in, but our learned internal information that's being projected onto the red in the context of looking at a traffic light makes us know that the red in this particular context means stop, as opposed to green, which means go. So that's kind of a way to think about top-down for bottom-up processing. So these pyramidal neurons are well situated to be able to negotiate these types of input. So there might be times in which we need to really process a lot of bottom information. We may need to turn off some of our top-down kind of processing, and sometimes we need to kind of amplify the top-down information to be able to provide more subjective interpretation of our of the experience that's coming in and around us. And so what we find, and I'll go into the evidence that shows this, is that one of the cell types, BAS1A, seems to operate as a circuit hub that really seems to be selecting for or triggering or biasing the network to bottom-up processing. And I'll explain how we found it came across this information. One of the things that we did is we tried to characterize the functional responses of these different types of excitatory neurons. What we find is the BAS1A cells are very highly active, and they also are very highly tuned to the Whisker tactile features. So they act as really a bottom-up, highly active feature detectors. The other thing that we looked at, and one of the things that we can leverage with the spatial transcriptomics properties, is the ability to look at multiple cell types and how they're interacting with each other. So we can look at and create functional networks of how different cell types really are really functionally interacting under the context of their behavior. Okay? So we can perform these GLM analysis. We can generate network graphs on a functional level where each of these nodes constitute different types of cells. And what we find is that the BAS1A neurons are highly orchestrated of local network activity. All the other neurons that we're imaging simultaneously are highly informed by the activity of the BAS1A neurons. In contrast, the inhibitory cell type somatostatin, these neurons are followers of network activity. they are the neurons that seem to be highly informed by all the other neurons around them. If we start to look at the different nodes and the strengths between these cell types, what we find is that what's reoccurring is there just seems to be strong functional connections between the BAS1A cells and the SST neurons. And so that suggests that potentially these functional connections are established by anatomical connections. So to verify this, we again leveraged spatial transcriptomics, but we integrated spatial transcriptomics with anatomical tracing. And so I don't know, people might or might not be familiar with monosynaptic rabies tracing or virus tracing, but just briefly kind of highlight how it works. It's a viral-based tracing in which you can infect in a genetically defined manner specific cell types and label them as starter cells. Then we can use rabies virus that will go in and infect these starter cells. And as the rabies virus replicates, it will jump one synapse back to label the inputs to these specific starter cells. And so this is a way to map synaptic connections between neurons and the networks, but it can be done in a genetically defined manner because you can, using conditional genetic approaches, label specific starter cells. So this is what we did. We went in and we used transgenic animals expressing Cre recombinase and somatostatin neurons. We infected the somatostatin neurons and labeled them as starter cells. And that's shown in pink here. And then we went in with rabies to infect these particular SST neurons. And all the green neurons that you see here are the neurons that provide input onto the SST cells. As a control population, we took another set of animals that expresses Cre using VIP neurons and then mapped their inputs. So we did this experiment, got the histology, and then we performed spatial transcriptomics to identify which are the excitatory neurons, are the inputs, right? So for every green neuron, we can stain and look for the fish markers. So when we do this, what we find is that the BAS1A neurons, specifically in the upper layers of layer 2-3, these are the neurons that seem to be providing preferential input onto SST neurons. And so this seems to confirm our, at an anatomical level, are what we've measured on a functional level. So to kind of put together a circuit diagram, what we see is that BAS1A neurons provide excitatory input onto SST neurons. And these SST neurons are providing inhibitory inputs onto the top-down apical inputs of other excitatory neurons. So you can imagine the situation if there's a lot of bottom-up information coming in, these BAS1A neurons are highly active, and then they will go and excite the SST neurons, which will then shut down the top part of the inputs to the neurons, to other neurons, which are receiving top-down input. And this will bias information that's being processed in the network towards the more bottom-up information. Okay, so this is one circuit that we identified using the CRACK platform. The other thing that we came across was also interesting, gets back at the idea of transcriptional states and whether or not the gene expression patterns that we're seeing in the brain are in fact static or dynamic, right? So as I mentioned earlier, one highly dynamic transcriptional state is the state that's related to learning and memory neuronal plasticity. There are a large number of these activity regulated or immediate early genes that go up and down with experience or activity that are essential for synaptic plasticity and neuronal plasticity. What we find, or what the Allen Institute found in their RNA sequencing studies, was that BAS1A neurons are selectively enriched in subsets of immediate early genes. So there are genes related to plasticities like FOS, for example, that seem to be upregulated in these particular cells and not other types of cells. Now, all of this kind of work was done in naive animals that were not trained to any behavior. So it suggests potentially that, kind of counter to what people think, potentially the elevated expression that you see in this particular cell type might not necessarily be a dynamic property, but in fact a stable property of this particular type of cell. So we really wanted to understand, well, what is going on with this expression and how does that relate to the function of the cell? So here we turn to animals, transgenic animals that others, specifically Alison Barth has created. So she has generated these mice that expresses the GFP under the FOS promoter and as a way to label cells that show high FOS expression. And one of the things that she's found when she recorded from these cells was that these cells were highly active and also highly responsive to sensory input. And so it's potentially the cells that she looked at, these fosGFP cells, constitute some subpopulation of neurons of these BAS1A cells that we've been studying. So to kind of look at this, we used her mice, and because we were looking at the FOS protein GFP, we can actually also track the FOS GFP dynamics in an awake animal using 2-photon imaging. So what we did is we took these transgenic animals, we monitor FOS GFP expression levels across time. We also express our calcium indicator so we can monitor neuroactivity. And we performed a experience-dependent plasticity paradigm, a sensory deprivation paradigm, which has been known to downregulate or change FOS GFP levels and also change neuroactivity patterns. But we wanted to ask, what happens in the BAS1A cells if we sensory deprive these animals? And so what we did was the following. We stimulated the whiskers to characterize the responses. Then we trimmed all the whiskers. So now the animals were no longer getting sensory input from their whiskers. And along this time, we imaged both the FOS GFP protein across this deprivation experiment and the activity pattern during the deprivation experiments. After the experiments were done, we took the tissue out and we performed crack to look at what types of cells we were looking at. So this is what we see. So we'll first focus on the activity and the FOS GFP dynamics. So when we trim all the whiskers, we see the activity of the area goes down. This is not surprising. It's been observed before, but there is a slight, after a few number of days, a slight homeostatic compensation recovery of the sensory responses after deprivation. If we look at the fosGFP expression, we can categorize cells as whether or not they're just never express fosGFP, whether they show dynamic expression of fosGFP, whether it goes up or down, or whether or not, cells that show high stable expression of fosGFP. And what we find is that the cells that show high stable expression of fosGFP are the ones that are specifically showing this homeostatic recovery in response to sensory deprivation. If we pair this with the cell type information, what we find is that if we look at the cell types and whether or not they're dynamically or stably expressing fosGFP, what we find is that when we trim the whiskers, the amount of dynamic fosGFP expression goes up. There's more turnover in the other two cell types, ADAM-TS2 and ASHMAT, but the amount of dynamics that we see in the BAS1A cells is consistent or the same as before the deprivation. So this reinforces the idea that the fosGFP, FOS expression in these BAS1A cells is a stable feature of this particular type of cell. And furthermore, if we look at how functionally the neurons change, the BAS1A cells are also the ones that show this homeostatic recovery. So something about the genetic program of these BAS1A cells has conferred a kind of high immediate early gene expression pattern. And it seems potentially that these cells are kind of primed to respond under changes in experience, potentially to maintain network activity patterns or certain representations when the animal is subjected to these new experiences. All right, so hopefully that kind of gives you a vignette of the types of things that we're kind of leveraging, things like spatial transcriptomics for. And I think it's just the beginning of really being able to think about questions in neuroscience that will allow us to really take advantage or really leverage this idea of more vertically driven approaches to combine both behavior, activity, connectivity, and gene expression within the same individual samples or specimens. And where do we go from here? And this is, great to talk to people, in this forum and more offline. Is on our end, I think there's a lot of things that we can do, and I think there's a lot of opportunities to explore. Given the current technologies that are available, which I'm seeing coming out every week, a lot of these things seem to be kind of dedicated towards more thin tissue sections, but it'd be great to think about ways where some of these approaches can be extended to more thicker sections, where again, it's more easier, more amenable to thinking to kind of these ex vivo, in vivo registration problems or challenges. Other things that I think are be interesting to kind of look into are things like integrating connectomics, right? I highlight using Rabies tracing, some ways to integrate some amount of anatomical tracing with both genomic gene expression patterns and neuroactivity, but there could be even more ways to do it in a more dense manner. And the last thing that I talked about when we were tracking fosGFP dynamics, right? It'd be great to look at a large number of other types of gene expression patterns, right? So right now we can kind of capture a snapshot, of gene expression patterns at the end of a given experiment. Our ability to look at markers longitudinally over the course of these imaging experiments is usually limited to maybe 1 or 2 markers, right? Are there ways potentially to multiplex this so we can look across multiple markers in a living animal? So again, happy to talk and brainstorm about these different ideas. And with that, I'd just like to thank all of you for your attention. So this was work carried out primarily by Cameron Connellis in the lab, who just graduated and has moved on, in collaboration with people from the Allen Institute, who really helped with providing us the initial RNA sequencing information and also helping with the rabies experiments. And yeah, if people in this audience are interested in any of these ideas or trying new ideas, we're looking to hire postdocs, so please come and get in touch. Thank you very much.
Yeah, thank you, Jerry. Very, very impressive, and I learned a lot on neuroscience. Yeah, I think in this field, I think the seminars we organized so far in the neuroscience topic, we really don't have anyone talking about the connectomic stuff. So this is, I feel this is kind of the missing piece on the gap right now. Yeah, how to bridge the omics, spatial omics, and the truly functioning data like connectomics.
Wonderful. From the audience, do we have any questions? Feel free to unmute yourself and also ask if you have. Otherwise, feel free to also post on the chat. So maybe while waiting, we can ask a question. So the multiplex gene expression technology is basically limited to gene in a thin tissue, right? Relatively thin tissue, and you're interested in going to the thicker. So in the data that you are missing the other part of the brain, do you think it's losing the connectomics, or like what you basically create here is a subset of the connections, or how do you, basically comment on this? How do you kind of visualize this?
So you're talking about like the rabies tracing here. Let's see.
Yeah, when you combine them, exactly.
Sorry. Yeah, so here we're also working with 150 thick micron sections. So it's a similar thickness, and so we're essentially, we are looking at more local connectivity. We're not necessarily looking at multiple sections, although that would also be possible, I think, to look at sections. And so yeah, so this is one way to look at connectivity. It's very it's targeted because we're looking at certain cell types and we're looking at inputs of certain types. It's not dense maybe, and I think there are other ways. I think other strategies that people are using that could allow us to look more densely. And the idea with the and maybe you guys know a little bit better. I think the main challenge, it seems to me, like the reason why people are sticking to thin sections is for two reasons, I guess. One is just registration. It just makes it easy. It's just easier to register if it's 2D versus 3D, right? And then the other concern is whether or not you're getting reliable penetration and staining, consistently when you go to thicker sections, right? And so again, I don't know, maybe there are ways, maybe people are working on this, I hope. But if there is, it'd be great to kind of see what might be possible in those.
I see. And you are taking these pictures in 40x or something, or what?
So all the kind of high resolution is with a 40x and a spinning disk confocal.
Wonderful. Yeah, it's beautiful data. I mean, we also work with image-based gene expression data, and this is really great. And the way that also you create barcodes is also very promising, even with HCR. And
yeah, I think it works again for targets that are where we are confident that are non-overlapping. There are cases in which if there's mixed, right? There are some neurons that kind of look that we can't figure out exactly what's going on because there's overlap, right, of some expression, and sometimes we can kind of zoom in and we can kind of see, disambiguate what's going on, but that it really, it would kind of really involves having to go in and look at the single molecule level. And so if there are ways to, it'd be great to open up the doors, right, to other markers that we are more gradient-like and show where the being able to quantitate, be more quantitative about the expression levels is also informative. Right?
Wonderful. Yeah, your binary separation was even very convincing. Multiple types expressing. And we have a question actually. Chang Wu Seo is asking how much of functional clusters defined by neural activity is explained by transcriptomic clusters? If you have info, how about connectivity clusters?
Yeah, so I think I think it's a slowly evolving story, but I think so far, which is kind of, as we start to break things down, we start to see functional segregation. And, we haven't started to, and I think the way we break things down so far is maybe just one level down, right? And if we keep breaking things down, the question is, will we continue to see functional segregation, right? And so I think that's the open question, right? So what we're seeing is so far as we're breaking things up, we can see functional clustering. And if we have a higher depth to be able to go even further, these are the kind of questions that we want to test, right? At what point do you go down and keep breaking things down and you still see segregation? Or at what point, maybe there might be a certain point in which you can't distinguish, right, functionally what's going on, even if you are able to kind of continue to break things down at a transcriptomic levels. And then in terms of connectivity, I think, right, there is some, it's my impression with the connectivity, there is going to be some differences in the rules, right? So on a gross level, there might be some segregation, right, or some separation. But at a fine scale level, because there's so much already known in terms of how activity and experience can change the wiring, a lot of the wiring probably has some amount of non-cell type specific or non-genetic rules. And so there's probably a layering of genetic rules that also kind of overlaps with non-genetic rules. And so what you get is more, maybe less discrete, but more kind of gradient and types of patternings.
Wonderful. Any other questions? Okay, so yeah, great.
Yeah, so Jerry, in one of the earlier slides, you show the calcium signaling you can. So in that case, can you see the signal propagation through the synapse axons? And I'm not familiar with in this field. So can you build some sort of connectivity map using the imaging alone?
Yeah, so you can express these indicators in subcellular compartments. And so a lot of what I showed you are maybe more in the head body or has a more larger fill, but you can express them in the axons or you can express them in the dendrites and you can see the activity patterns. And so one thing that people can do, for example, is inject the indicator in one area, the axons will go into a different area. And when you image that area, you're only imaging the axons. So you both see on a subcellular level the activity patterns and maybe it's the connectivity, but you also have an awareness of where those neurons are coming from, right? And they're not coming from other neurons. And so again, one open question, which I think maybe you guys are handling or maybe tackling is, we're just looking at transcripts in the cell body, right? But there's probably transcriptional machinery, transcripts in the subcellular compartment of the axons or the dendrites that might have some synaptic specificity, right? It'd be great to start to look at these, subcellular compartments and see what's going on.
Actually, I remember earlier papers that they observed some RNA clusters, near in the synapses and stuff like that. And that's actually a classical example of spatial, biology being like asymmetric and RNA being non-pose undistributed, right?
Yes, there's definitely a lot of post-transcriptional, transcriptional local translation, right, of things that are happening, right? Because of course, if the axons can traverse a very long distance, right? And there's probably things going on at the very end of the axons in a different area that are having certain effects, right, on a long range scale.
Exactly.
Yeah, also in this case, KKO do electrical reporting and then kind of take that brain section out and correlate with the spatial transcriptome.
Yeah, so I think there's two things. So one of the things that we're pushing on is not calcium imaging, but voltage imaging. So there we can have more direct readout of electrical activity.
Okay.
And so that's one approach I think should be promising or really add another level to some of these types of combined physiology and gene expression experiments. We haven't done anything in terms of like patching, although there are many other groups who do like
patch sick
stuff like that. We haven't done anything like that, but I think that's definitely a very powerful approach. It's just a little bit more limited because you only get a couple of cells at a time. It's difficult to get a lot of cells, right? For them.
Wonderful. It's about 3:03. I mean, we can maybe have one more question. And also the field currently that they are using spatial transcriptomics, not directly imaging, right? What do you think the overall main difference between the two?
There's been a couple papers recently there. So I think there's been a growing interest in doing both the imaging and the transcriptomics. And there are more and more groups doing it. So I anticipate that's going to be more and more, more and more common. I think, one of the areas I think it's really starting to kind of really take off is, we're working in mouse models where things like imaging and functional imaging and these things are very easily to do. But I think extending this type of spatial transcriptomics to like human samples or non-human primate samples, or model organisms, I think that's really where, in my impression, I think there's going to be an explosion of knowledge that can be gained by starting to look at different types of, non-genetic model organisms.
Wonderful. Thank you. And with this, thanks to Professor Chen again. I'm going to use this clapping sign for this wonderful talk. And I appreciate it, taking time and presenting the beautiful data. And before we wrap up, I'd like to also announce the next week's speaker. That is a PhD student from Dr. Jian Ma's lab from Carnegie Mellon University, and his name is Tianming Zhou. And he will talk about SpiceMix. This is a computational method to integrate single-cell spatial modeling for inferring cell identity. With this, thank you, everyone, and hope to see you guys next week.
Thanks so much again. See you later.
Bye-bye.
Thank you, Jerry.
Thank you.